RNA. As you read English, do you read DNA? Genes in the DNA contain coded instructions that tell cells how to build proteins. The first step in decoding these genetic instructions is to copy part of the base sequence from DNA into RNA. RNA, like DNA, is a nucleic acid that consists of a long chain of nucleotides. RNA then uses the base sequence copied from DNA to direct the production of proteins. Each nucleotide in both DNA and RNA is made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base. There are 3 important differences between RNA and DNA. The sugar in RNA is ribose instead of deoxyribose. RNA is generally single-stranded and not double-stranded. RNA contains uracil in place of thymine. These chemical differences make it easy for an enzyme in the cell to tell DNA and RNA apart. The roles played by DNA and RNA are similar to the master plans of blueprints used by builders. A master plan has all the information needed to construct a building. Builders never bring a valuable master plan to the building site where it might be damaged or lost. Instead, they prepare inexpensive, disposable copies of the master plan called blueprints. Similarly, the cell uses DNA master plan to prepare RNA blueprints. The DNA molecule stays safely in the cell's nucleus, while RNA molecules go to the protein building sites in the cytoplasm, the ribosomes. You can think of RNA molecules as a disposable copy of the segment of DNA, a working copy of the single gene. RNA has many functions, but most RNA molecules are involved in protein synthesis only. RNA controls the assembly of amino acids into proteins. Each type of RNA molecule specializes in different aspects of this job. The 3 main types of RNA are messenger RNA, mRNA, ribosomal RNA, rRNA, and transfer RNA, tRNA. Here's mRNA with the uracil in place of thymine. Here's the ribosome, is ribosomal RNA. And then the last one, transfer RNA with an amino acid attached to 3 bases. Most genes contain instructions for assembling amino acids into proteins. The RNA molecule that carries copies of these instructions are known as messenger RNA, mRNA. They carry information from DNA to other parts of the cell. Half a helix, not a double helix. Proteins are assembled in ribosomes, small organelles composed of 2 subunits. These ribosome subunits are made up of several ribosomal RNA, rRNA molecules, and as many as 80 different proteins. When a protein is built, a transfer RNA, tRNA molecule transfers each amino acid to the ribosome as it's specified by the coded messages in mRNA. It's a bit of a package with 3 bases. How does the cell make RNA? Most of the work of making RNA takes place during transcription. During transcription, segments of DNA serve as templates to produce complementary RNA molecules. The base sequence of the transcribed RNA complement the base sequence of the template DNA. In prokaryotes, RNA synthesis and protein synthesis take place in the cytoplasm. In eukaryotes, RNA is produced in the cell's nucleus and then moves to the cytoplasm to play a role in the production of proteins. Our focus will be on the transcription of eukaryotic cells. Transcription requires an enzyme known as RNA polymerase that is similar to DNA polymerase. RNA polymerase binds to DNA during transcription and separates the DNA strands. RNA polymerase then uses one strand of the DNA as a template from which to assemble nucleotides into a complementary strand of RNA. So the RNA polymerase uses this to create the mRNA. RNA polymerase binds only to promoters, regions of DNA that have specific base sequences. Promoters are signals in the DNA molecule to show RNA polymerase exactly where to begin making RNA. Similar signals in DNA cause transcript to stop when a new RNA molecule is completed. RNA molecules sometimes require bits and pieces to cut out of them before they can go into action. The portions that are cut out and discarded are called introns. Here's a piece of intron. Here's an intron. They are removed to make the full mRNA without those two intron pieces. In eukaryotes, introns are taken out of pre-mRNA molecules while they are still in the nucleus. The remaining pieces, known as exons, are then spliced back together to form the final mRNA. Exon parts that we keep for the final mRNA. Biologists don't have a complete answer as to why cells use energy to make a large RNA molecule and then throw parts of that molecule away. One consideration is that some pre-mRNA molecules may be cut and spliced in different ways in different tissues, making it possible for a single gene to produce several different forms of RNA. Do any intron or exon incorrect exchanges have potential to cause mutations? Do they have potential to change future generations?